All right, class. Now, let's do one last thing about action potential before we move on to reviewing the PowerPoints. So remember, the sodium gates or the voltage gated sodium channels, they have two gates, right? So let's draw it out. So this is one gate, and then the ball and chain thing is another gate. So this is when the voltage gated sodium channels are closed. This is closed state. Now when they open, this gate opens and this chain remains like this. So this is also voltage gated sodium channels, and this is open. When it's open, sodium can rush in. And then there is this another state where this gate is open. However, the ball blocks the gate, right? So if I go back, where was that? Right here. Do you see that? So this is closed, where the gate is closed, open, and then this is inactive state. So, and we drew all these here. So we have closed, open. Oh, I forgot to do this. Voltage gated sodium, and this is inactive state. Okay. So during graded potential, what do you think, in which state are the voltage gated sodium channels? Are they open, are they closed, or are they inactive? Well, during graded potential, the voltage gated sodium channels are closed. Only the ligand gated sodium channels are open, right? So at this point, they are like this. How do these voltage gated sodium channel look during depolarization? So this is depolarization, and from threshold to the peak, which is positive 30, during this phase the voltage gated sodium channels are wide open and sodium can rush into the cell. Okay, what happens here? At the peak, the voltage gated sodium channels are deactivated. Sorry, they are inactivated. So here they are inactive, open. Okay, now listen carefully. During repolarization, as more and more voltage gated potassium channels open up, these inactive voltage gated sodium channels reset. So during repolarization, the inactive voltage gated sodium channels reset themselves back to closed state. Okay, so one more time, let's review. We know that the voltage gated sodium channels have two gates, right? So when they are closed, it looks like this. When they're open, both gates are open, and during inactivated state, the gates are closed by the little ball. Okay, so during graded potential, these channels are closed. During depolarization, they are wide open, so more and more sodium can rush in. They open, more sodium. Again, more sodium voltage gated channels open, more sodium come in. So it's a positive feedback type of thing going on during depolarization. These gates get inactivated at positive 30. So at positive 30, the ball kind of blocks these, and this state is inactivated state. And during repolarization, the inactivated voltage gated sodium channels come back or reset themselves back to their closed state. Okay? Why aren't we talking about the voltage gated potassium channels? Well, remember the voltage gated potassium channels, they just have one gate. So they either open or close. When they open, then we have repolarization, and when they close, they are closed. Nothing can pass through. Since the voltage gated sodium channels are a little more complicated with two gates, I thought it's really important to understand how they behave during the action potential. All right, let's talk about let's go through the PowerPoint and make sure we completely understand action potential. So I'll just do slideshow now. All right, class. So on the right hand side, we do see the diagram of the action potential. We do see that graded potential in pink, which is the depolarization from -70 to -55. So this right here is graded potential. Okay, now what happens at threshold? At threshold, which is -55, voltage gated sodium channels open up and sodium rushes inside the cell. More sodium comes in, more depolarization. And that causes an action potential. When the inside of the neuron is depolarized enough, then at the axon hillock, a wave of action potential is initiated, right? So as the cell depolarized, more channels open, and this is like a positive feedback loop. And this causes an overshoot of the membrane potential to +30, as we see. And remember, membrane potential reaches +30, and this is depolarization. So when sodium is in, do you see number 1, sodium in? This whole part is depolarization. All right. So what happens at +30? At +30, as we mentioned before, the sodium channels close. Well, it's not 100% correct to say they are closed. Know that they are inactive. They are inactivated. And the voltage gated potassium channels open. So voltage gated potassium channels open at this point. And as these channels open, potassium rushes out. And that results, so we are, as potassium rushes out, we are losing positive charge, and that brings it, and that is responsible for repolarization of the membrane. This is a negative feedback loop. Okay, so the potassium channels, the voltage gated potassium channels, as they open, obviously potassium leaks out. This is called repolarization, and the membrane potential comes back to -70, right? But there is another state called after hyperpolarization, where the repolarization just doesn't stop at -70. It kind of overshoots and goes down to -85. Do you see here? It doesn't stop at -70. It continues down to -85 millivolts. And this is because the leak channels are still open. And it is the sodium-potassium pump and the activity of the leak channels, everything together, bring the resting membrane potential back to -70. So again, remember, in this state, when it's hyperpolarized, it's the sodium-potassium pumps that are mainly working to reset the membrane potential back to -70. Obviously, the leak channels are always and always open. All right. Now, action potentials follow all or none law. What do we mean by that? Okay, so that means once a membrane potential changes from -70 to -55, boom, you will have an action potential. Now, is it possible that it takes a lot of time to reach from -70 to -55? Yeah. Is it possible that the threshold is reached super fast? Yeah. My point is, no matter whether graded potential takes place slowly or fast, once you hit the threshold, there will be an action potential. So let's talk about this diagram, which is on the right. Okay, so we do see that the resting membrane potential is at -70, right? And we have these red arrows showing different stimuli. Now, the weakest stimuli is on the left-hand side, shown by the short red arrow, and the strongest stimuli, or the strongest stimulus, is on the right-hand side, shown by a long red arrow, right? And by stimuli or stimulus, we just mean single quick shocks. Now, let's look at the weakest stimulus. The weakest stimulus is not producing any action potential. Why not? Because it's so weak that it cannot reach threshold, right? Now let's look at the second one. So this cannot generate an action potential because threshold is not reached. What about the second one? Well, the second one is strong enough that the threshold is reached, and let's say this is -55 in red. And once threshold is reached, obviously you have an action potential. How about this one? How about this one? Well, the stimulus is strong enough to overcome the gap, or the stimulus is strong enough so that the threshold can be reached. And as it hits threshold, boom, you have an action potential. And obviously, the strongest stimulus, you will also see an action potential. Now, okay, what's the point here? Look at the sizes of the action potential. Look at the three action potential, the size. This, this and this. Are they of same sizes? Yes, they are. So, if we change the strength of the stimulus, will the size of action potential change? I repeat myself. If I change the strength of stimulus, will that change the size of action potential? The answer is no. Size of action potential will not change based on the strength of stimulus. Okay, so that is what this diagram is trying to show. So let's read through. Once threshold has reached, an action potential will happen, no matter how strong or stronger or strongest the stimulus is. The size of the stimulus will not affect the size of action potential. The size of action potential will remain the same no matter what. And it will always reach +30. It will never go beyond +30. So the size of the stimulus will not affect the action potential duration or size. That's the most important concept. I'll stop here and then start with a new slide.